to this second Journal Club of the Online Integrative RNA Biology Community of Special Interest, or iRNA COSI. My name is Mathieu Canal-Vaillard. I'm a postdoc in Joseph Brasch and Kristen Lynch's groups at the University of Pennsylvania. The OI-RNA COSI is a community of researchers who use and develop novel technologies and computational tools to do RNA biology research. So if you're doing that kind of research or you're interested in that kind of research, you belong in this community. We want to hear from you. If you have ideas about how we can make the community grow, how we can help you network with other members, we're here to promote your research, help you reach other members. There are several channels that you can use to get in touch. We have the website, irnacosi.org. We have a Twitter account. We also have an email account that you can use to get in touch directly with us. This journal club is supported by the International Society for Computational Biology and the RNA Society. Both societies will be holding their annual meetings online this year. The ISCB will be July 13th to 16th, and you can still submit abstracts for this meeting. And the ISCB comprises the iRNA COSI sub-meeting, which is where computational biologists doing RNA research gather and share novel research. So this should be a very exciting meeting. You can get more information about the meeting at irnacosi.org. And the RNA Society meeting will be happening in just a few weeks from now, May 26th to 31st. So I encourage you to look these meetings up and register. Before we move to today's article, I am happy to announce what the next paper that we will discuss will be and who our next guest will be. And that will be Masa Ganbari from Uwe Ohler's group at the Max Delbrück Center in Berlin, who will be presenting her recent genome research paper entitled Deep Neural Networks for Interpreting RNA Binding Protein Target Preferences. So this next journal club will be on Tuesday, May 19th at 11 a.m. Eastern Daylight Saving Time. So we're staying to the same time slot on the Tuesday for now. This journal club will be hosted by my colleague Anupama Jha, who is a grad student with Joseph Brasch. So this should be very interesting. Now, today we have Ana Fiszbein with us, who is a postdoc in Chris Burge's group at MIT. Ana, welcome.
Thank you.
How are you this morning?
I'm okay, pretty good. How are you?
Yeah, I'm good. I'm very excited about this journal club. I have to say that the first journal club that we had a month ago went very well. We had Furkan Fazal with us, who introduced us to Apex-seq. It was a great journal club. We had lots of attendees, many questions. We also have quite a few attendees today. We have 45 people currently logged in. I would encourage all of you to ask questions on the fly. So if you see during the presentation something that comes up and you have a question about the experiment, about the interpretation, about the results, please use the Q&A section that you should find at the bottom of your screen to post your questions. I will be moderating the questions, taking them and asking them directly to Ana. I might save some of your questions for later during the presentation or at the end of the presentation, because we'll have more time for questions then. But because we wanted to be just like a normal journal club that we would have in a small room with just a few people with friends, just please go ahead and ask questions as they come. And we'll try to make it, as open as possible. It worked very well the first time, so I'm looking forward to this. I think it's a very interesting paper in any case. So I'm sure it's going to be a lot of fun. I don't think I have anything else to add at this point. You can also use the chat. I mean, if you have questions that you want to ask to other attendees, or you want other attendees to answer a comment, please go ahead and use that function. So for now, so the format of this journal club, of course, now we have the first author presenting their own paper. That's how we want it to be. So today, any question you might have, Anna will have the answer to your question, and that's amazing. That makes it a very fun journal club. Anna's smiling like it's possible that she won't have the answer. I don't think it's possible. So yeah, here we go. Anna, I'll leave the screen to you. And you'll see me coming on whenever there is a question coming for you.
Awesome. Okay, so I will share the screen right now. Can you all see the presentation?
Yeah, looking good.
Yeah, okay, awesome. Okay, so thanks so much for the introduction. And thanks for the opportunity to having me here today and present my work. So today I will talk about our recent work on exon-mediated activation of transcription stops. So as you can imagine, I'm very interested in gene regulation. When and why our genes are turned on and off. And I think this is such an exciting topic because it's really crucial for every aspect of cellular function. So gene regulation defines cell identity during development and cell differentiation. It is crucial for evolution and disease. It contributes to the 10 million cancer-related deaths each year. And understanding the regulation of gene expression might open up new strategies and possibilities for therapeutic manipulation of gene expression. So this is how we used to think of the directional flow of genetic information. Each step in gene regulation can regulate downstream steps. However, we now know that, for example, RNA processing events can feedback and regulate transcription. And we also know that protein levels and protein translation can also impact back on RNA stability and chromatin structure. And moreover, we used to think of a very simple structure of the mammalian gene architecture. We used to think that we have a few exons here represented by boxes, a few introns here represented by lines, and a few non-coding regions or UTRs. However, we now understand that most human genes have alternative promoters, undergo internal alternative splicing, and have alternative polyrylation and termination sites. So this much more complicated gene architecture enables the production of multiple RNA and protein isoforms that greatly expand the coding capacity of our genes. So let's focus on, let me first remind you what is splicing and how it can lead to alternative splicing. So splicing is when introns are removed and exons are joined together. Here we're seeing two components of elements of the spliceosome components, two spliceosome components, sorry, and recognizing the 3' and the 5' splice sites. So the spliceosome recognizes the splice sites, and depending on the strength of those splice sites, some introns can be retained, and some exons are sometimes included and sometimes excluded in the mRNA. And that's what we call alternative exons or skipped exons. So let's focus now on the life of one of those mRNAs. So as I said, most human genes have alternative promoters. So the first decision in the life of an mRNA is what is called the promoter selection. So transcription can start in one promoter or the other. After that, we have transcription initiation, transcription elongation. Then introns are removed and exons are joined together doing splicing. And we have binding of RNA binding proteins. And actually, these RNA binding proteins can bind at any time during the cycle of the life cycle of an mRNA. But the binding of RNA binding proteins will regulate different processes, as for example, splicing. After that, we have termination and polyrylation. Then the mRNA is exported into the cytoplasm. It's translated into a protein and degraded. But most importantly, all these RNA processing events here are mostly co-transcriptional, leading to the possibility of functional and temporal connections between transcription and splicing. This is one of the first compelling evidence of a co-transcriptional splicing. It came from Drosophila embryonic units, where we see that here, introns in yellow are looping into these purple transcripts even before those transcripts are released from the black DNA. So for the first time, we can visualize it here that splicing is happening at more or less the same time as transcription. So we can think that transcription and splicing are coupled. So we do know that transcription can regulate splicing. And we learned this mostly from two different models that has been proposed for some mechanisms explaining how this might be possible. So these two different models that I will briefly explain are definitely not mutually exclusive, and they can work together. And they have many exceptions. So one of the first models that was proposed to explain how transcription can regulate splicing, it's called the recruitment model. And so in this model, it shows that, for example, the C-terminal domain of the polymerase can act as a binding platform for splicing factors that then will travel along with the polymerase and affect downstream alternative splicing choices. Moreover, the second model that has been proposed, it's called the kinetic model. And in this model, we see that the elongation rate of the polymerase can affect also downstream alternative splicing choices. So we have here in red an alternative exon or an internal skipped exon. So the splice sites of this alternative exon are weaker than the splice sites of the flanking constitutive exons. So if the elongation rate of the polymerase is very fast, then both the weaker and the stronger splice sites will be exposed to the spliceosome at more or less the same time. So that favors more exon exclusion. On the other hand, if the elongation rate of the polymerase is very slow, then we will have transcription of the weaker splice site here, even before the stronger splice site has been transcribed. So that will favor more exon inclusion. Also, and more recently, we start thinking that the other way around is also true, and that splicing can feed back to transcription. And we learned this mostly from this phenomenon that is called intermediated enhancement of gene expression, in which introducing an intron in an intronless gene can increase gene expression levels. And this is why most of our expression plasmids have introns there. So when we started this project, my main question, what drives most of this research, was if exon splicing now, not intron splicing, can also feed back to transcription and affect gene expression levels. And what about multiple promoters? As we said, most human genes have alternative promoters. So what happened in a gene with multiple promoters and skipped exons? Could splicing of those exons impact the usage of alternative promoters? So those were the questions that I was trying to address. So for that, we developed a system and using thousands of evolutionary new exons that were recently identified using RNA-seq data across multiple species and multiple tissues. This is a pattern of inclusion of a mouse-specific new exon that is included in any of the 9 tissues that we sequence in mouse, but is never included in any of the tissues of the other species. And these alternative evolutionary new exons tend to be no-coding, located mostly in the 5' UTR, are usually alternative and tissue-specific. This is the pattern of inclusion of a rat-specific loss exon that is included in all other species, in any of the 9 tissues that we sequence in all other species, but is never included in rat. So my first observation was that most genes that has evolutionary new exons have increased gene expression levels. And we see that with the cumulative fraction of genes comparing the gene expression levels in mouse compared to rat. So you can see here that genes with inclusion of evolutionary new exons have increased gene expression levels. But, and this is important, only in the tissues where the new exons are included. On the contrary, we see that genes that lost evolutionary new exons in rat have decreased gene expression levels in rat compared to mouse. And we will see this for all different species that gain new exons or that lost new exons. So it's definitely not species specific. So this means that inclusion of evolutionary new exons is associated with an increase in gene expression levels. However, to understand, Matthew, you have a question?
Well, I do have a question. You're saying that this is something that you see not specifically in mouse, but also in other species. Can you comment specifically about human? How many of those exons you might find, and whether the trend is just as strong or stronger?
Yeah, no, that is a fantastic question, and I'm working on that. When I say other species, I'm referring to 5 species that we use for these particular work, and that not include human. So I'm actually working now on different tissues in human, and hopefully I will can get back to you in a few months to tell you what's going on with humans.
Right, right. Yeah, I think it's going to become especially relevant towards the end of the talk. So maybe we can come back to it later.
Yes, exactly. Yes, I will come back to that. And I have some data on humans as well, but not on evolutionary new exons in human, not yet. Okay, so this means that gene expression is an increase in gene expression is associated with inclusion of evolutionary new exons, but we didn't know which layer of gene expression was regulated. So to study that, I analyzed nascent RNA levels in studying pro-seq reads across different species. And what I observed is that genes that has mouse-specific new exons have increased nascent RNA reads or nascent pro-seq reads. That is a way to measure nascent RNA. So this points out that the effect of the increase in gene expression levels, it's based on an increase in transcription levels. However, this positive association between an increase in transcription or an increase in gene expression levels and inclusion of evolutionary new exon is only observed for those genes that have multiple promoters or multiple transcription start sites or TSSs. So I wonder, what is the distribution of these TSSs or transcription start sites in genes with evolutionary new exons? And you can see here the distribution of the number of TSSs for all genes with evolutionary new exons. So you see here how the fraction of genes with 1 TSS, 2 TSS, or more TSSs. And the important part here is that genes with mouse-specific new exons, the distribution of the number of TSSs in genes with mouse-specific new exons is shifted to the right. So genes with new exons have increased number of TSSs. And moreover, the gain of a new TSS is associated with the gain of an evolutionary new exon is associated with the gain of new TSSs. So here it's important to point out that we see a stronger effect when those evolutionary new exons are located towards the 5' end of genes, and in particular in the 5' UTRs. And also we observe an increase in gene expression levels species specific for those genes that gain new TSSs in that same species. So that means that the gain of the increase in gene expression levels or transcription levels by the inclusion of evolutionary new exons could be mediated by activation of new TSSs in those same genes. So a natural question that arises at that point is where those new TSSs are located. So we have the location of the evolutionary new exon here in red as the 0 coordinate. So we can think that those new TSSs can be located upstream or downstream of the new exon. And what we really want to know is where the location of those TSSs compared to the location in other species. So here I am showing you the distribution of the location of all different TSSs in mouse and in rat for genes with mouse-specific new exons or the homologous genes in rat, always relative to the coordinate of the position of the new exon here in 0 in the mouse genome or the homolog coordinates in the rat genome. So you can see here that those new TSSs in mouse peak right upstream, or sorry, just peak that are centered around the position of the new exon. But now, if we zoom in, we see that those new TSSs in mouse peak right upstream of the position of the new exon within a window of 1 kb here. So these TSSs arises right upstream of the position of the new exon, and I guess there's a question.
Well, I do have a question. So you're centering your plot around the new exon, which is not included in rat, but it's conserved in rat, and it's just a weaker splice 3' splice site, or can you explain how that happens?
Yeah, no, that's a great question. So those mouse-specific new exons are only included in the mouse transcripts. They're never included in rat. So that means that if the splice sites are kind of conserved in the rat genome, they are like definitely super weak because they're never included. So what actually most of the time happens is that one of these splice sites are not conserved in the rat genome. So,
but generally speaking, the sequence of the exon itself is conserved?
Generally speaking, these genes are very conserved between different species, and we're going to see that later on. But more or less, the sequence upstream and downstream of the exon are usually conserved. The sequence of the exon itself, it's pretty well conserved as well. However, most of the time there can be a deletion in the upstream intron. And often we see that one of the splice sites are not conserved in the other species. Great. So the gain of a new splice site in the mouse genome drives the inclusion of these evolutionary exons.
Great, thank you.
So we also see that those TSSs that arises here upstream and proximal to the position of the evolutionary new exons have the strongest connection with the usage of these evolutionary new exons. So here I'm showing you the correlation between the inclusion levels of these mouse-specific new exons and the usage of each TSS on the same gene, depending on the location or the relative location of the TSS relative to the position of the new exon. And what we see is that those TSSs located right upstream and proximal to the position of the new exon have the highest positive association. So until now, I have shown you that there is a connection, there is an association between the inclusion of evolutionary new exons and increased gene expression levels, and also an association between the inclusion of these evolutionary new exons and the usage of proximate and upstream transcription start sites. But now I'm going to show you with experiments that the inclusion or the splicing levels of these new exons can really drive or activate cryptic promoters located nearby. So starting with experiments, we focus on these two candidate genes that has very strong connections between gene expression levels and the inclusion levels of mouse-specific new exons here, always compared to other species. So I use antisense morpholinos to block the splice sites of these evolutionary new exons. And when I use these morpholinos that block the 5' or the 3' splice site of the evolutionary new exon, we see a downregulation in the inclusion of the new exon, and that is followed by a downregulation of nascent RNA levels. Also pointing out this regulation of splicing on gene expression levels, and in particular on transcription levels. So using this other candidate gene with CRISPR, I mutate the splice site of this evolutionary new exon in this other gene. And I saw that when I knock out the inclusion of this mouse-specific new exon, we see a downregulation in the usage of the upstream TSS and an upregulation in the usage of the downstream TSSs. So you can see here that here we're measuring nascent RNA levels as well. So we think that this upregulation in the downstream TSSs is something specific about this particular gene, because we didn't observe that in other candidates. But what is very consistent is the downregulation in this upstream TSS. And what is more important here is that we observe the same effect on the sense direction than in the antisense transcription. And this is important because it points out that splicing of these evolutionary new exons can impact transcription in both directions, but will not impact the directionality of these promoters. So we wanted to go farther, so we used this other candidate gene that is called TSQ or TSQU that has one mouse-specific new exon here and two upstream TSSs, alternative TSSs. So in mouse, both TSSs are used at similar levels. And here I'm showing that with 5' rays. However, when I use an antisense morpholino that blocks the usage of those splice sites, we see a shift towards the usage of the more distal TSS and a downregulation of the proximal TSS. And this is a relative measurement. But the downregulation in the proximal TSS is also seen by a decrease in H3K4 methylation levels that is a mark associated with transcription initiation and promoter activity. But we wanted to go a little further and ask if the inclusion of these evolutionary new exons can be sufficient to activate a cryptic TSS located nearby. So to study this, I use the genome, the rat genome. So in the rat, as we discussed, that happened in most of the cases, the sequence is pretty well conserved compared to the mouse sequence. There is a 5' splice site in the homolog coordinate of the mouse-specific 5' splice site for the mouse-specific new exon. However, there is not a 3' splice site. So this exon is not included in the rat genome. 5' rays shows that this transcript in the rat sequence, it's driven by the usage of the distal promoter. And in the context of a plasmid, we do see some basal levels of transcription from the upstream TSS. But in the rat transcriptome, we mostly don't see anything coming from this TSS. However, this TSS, the sequence, is very well conserved between mouse and rat. So what I did was to create a 3' splice site of the mouse-specific new exon, now in the rat context. And when I create a 3' splice site, we see an inclusion of the mouse-specific new exon now in the rat genome. And when we did that, I observed an activation of the predictive TSS that was located nearby, and also an increase in transcription levels or gene expression levels, because the usage of this promoter is not well affected by there is an increase in the usage or an activation in the usage of the cryptic TSS located nearby. And I also know that this effect depends on splicing and not in the presence of a 3' splice site, because we mutated this 3' splice site and not only to be the mouse 3' splice site, but with a stronger 3' splice site. And I also mutate the 5' splice site. And I only see, or I only observe this activation of the cryptic promoter when both the 3' and the 5' splice site are present and this exon is included. So to try to understand a little bit of the mechanism that can be driving here, there is a question before that.
Yeah, before we move on, so I have a question from Amar, who asks, what are the general features of these novel exons? And I think you're about to get at that question, but more importantly, most of these novel exons are located in the 5' UTR. Is that correct?
Yes, that's correct. So most of these evolutionary exons are located in the 5' UTR, and so a few are not located in the 5' UTR, but we see these effects are stronger for those located in the 5' UTR. So yes, we're getting to a key point where it's important to have this the splicing of these exons located nearby of the position of these cryptic promoters.
Right. And also another question that Amar had that's probably more difficult to answer with the kind of data that you have, but the question is, are these novel exons co-regulated with other splicing events elsewhere in the transcript?
Wow, that's a fantastic question. I guess the answer is yes. I don't know. I don't have much data to show here, but I do see a correlation between the usage of these evolutionary exons with, well, of course, promoter, that's what we're talking here, and associated first exons. But I also see a connection with inclusion of other skipped exons in the gene and the usage of alternative last exons as well. And that's part of what we're working right now to try to understand that better. But that is a fantastic question. I'm very interested in that.
Great, thank you, Anna.
Sure. Okay, so we are trying to understand a little bit of the mechanism here. So for that, I analyzed RNA-seq data from a recent ENCODE project where they deleted or downregulate the usage or the expression of hundreds of RNA binding proteins. So I was trying to analyze this data to assess the changes in TSS usage or promoter usage by depletion of these RNA binding proteins. And I surprisingly found that most RNA binding proteins affect the usage of alternative promoters. And I selected the 10 splicing factors here in red, 10 splicing factors that drive the largest changes in TSS or promoter usage. And I built a protein-protein interaction network. So here we see those 10 splicing factors driving the largest changes in TSS usage. And those 10 splicing factors include PTBP1 and HRPU and many others that you probably know. But what is important right now is that those 10 splicing factors interact with other 65 different proteins. And those proteins are involved in transcription from RNA pol2, in enhancer binding, in promoter binding. There are usually transcription factors or transcription cofactors. And here, actually, we can see some subunits of RNA pol2 here and some core transcription factors. So we think that these splicing factors co-associated with transcription factors. And to test a recruitment model in which splicing factors can recruit transcription factors locally, I use the case of HRPU. And so with Keegan, we work on HRPU that has one RNA binding domain towards the C-terminal domain of the protein and has connections with other proteins towards the N-terminal domain. So in particular, HRPU interacts with this core transcription factor called TF2F. That's one of the transcription factors that we saw in the protein-protein interaction network by its N-terminal domain. So HRPU can regulate the splicing levels of the mouse-specific new exon in TSQU. So downregulation of HRPU decrease because it binds close to the position of the new exon. So a decrease in depletion of HRPU decrease the inclusion levels of the mouse-specific new exon in TSQU, and we do see a decrease in TF2F recruitment levels. If we overexpress a full length of HRPU, we see a rescue of the splicing phenotype and also a rescue in TF2F levels. However, if we overexpress a truncated version of HRPU that only contains the C-terminal domain and lacks the N-terminal domain, we see a rescue in the splicing phenotype, but this fails to rescue TF2F levels. So this experiment shows an example of where the recruitment of this splicing factor, HRPU in this case, can be mediating the splicing-dependent activation of this critical promoter or critical TSS by recruitment of TF2F. I guess if there are no questions here, I will continue. So we see, and what I've shown you until now is that during evolution, there is mutations that can create splice sites. The creation of new splice sites increase the inclusion of evolutionary new exons. But the inclusion of these evolutionary exons recruits spliceosome components and splicing factors, as I show you with the example of HRPU. But we believe that these splicing factors are not coming alone, and they interact and they co-associated with transcription factors as well. So now we have a high concentration of transcription factors close to a position where there might be a cryptic promoter or a cryptic TSS. So this high concentration of transcription factors can be sufficient to activate cryptic promoters located nearby. And once activated in this way, these new promoters generate new mRNA isoforms and an increase in gene expression levels of those host genes. So the natural question that arises at this point, and something that most of you were wondering, is what is special about these evolutionary exons? And the answer to this question is nothing. There is nothing special about evolutionary exons, and we see that EMATS, that's how we call this, or exon-mediated activation of transcription starts, can affect thousands of mammalian genes. Here I'm showing you just one example of one gene in mouse that has 3 alternative promoters, 3 alternative associated first exons, and then 2 skipped exons that are conserved skipped exons that didn't recently evolve. They're like normal skipped exons or alternative exons. And what we see here is that depletion of this highly included and proximal skipped exon have an effect on the usage of all 3 alternative promoters. So downregulation of the inclusion of this highly included proximal skipped exon downregulates all 3 promoters with the largest effect for those promoters located nearby. And we can see that here. Downregulation of the inclusion of this medium included skipped exon and located more distal has also an impact in this proximal TSSs, although the effect on this distal TSS is not significant, and this effect here is smaller. So this points out that the splicing of the skipped exons can really regulate the gene expression levels and the usage of alternative promoters here. And as exemplify here, but also looking in a genome-wide scale, I found that the strongest effects of EMATS are for those weaker promoters when they are located upstream of a highly included skipped exon. So here you can see the highest effects of this activation of cryptic promoters are, or promoters not necessarily cryptic, when these TSSs are located right upstream of the position of a highly included skipped exon. So this really points out that there's nothing specific about new exons, and that EMATS can affect thousands of genes where we see weak TSSs and skipped exons. So as I said, looking in a genome-wide scale, we found that EMATS affect thousands of mammalian genes, now in the human genome as well. And we found the strongest effects when there is a highly included skipped exon, when there is an intrinsically weak transcription start site located upstream, and when they are in high proximity. I mean, when the weak TSS is located between 1 and 5 kbs upstream of the position of the highly included skipped exon. So here we have a list of what we call EMATS genes, or genes with the EMATS structure. And we have the numbers here for mouse. So we see almost 4,000 of genes that has a weak TSS located upstream of a highly included skipped exon. And we have less than 2,000 genes in which that weak TSS is located less or within a window of 2 kbs upstream of the position of the skipped exon. So for human, the numbers are pretty similar, and that's where we're seeing some of the effects in human as well, but we're not talking about evolutionary new exons here, but we're just talking about genes with what we call an EMATS structure. And something that you might be thinking about is what happened with constitutive exons, right? So if we take to a limit a highly included skipped exon, that would lead us to constitutive exons, right? So if we consider constitutive exons, the numbers really increase. So there are almost 6,000 human genes, what we can consider genes with an EMATS structure. And yes, there is a question here. Yeah.
Yes, there's going to be a question. So by the increasing the inclusion of those novel exons and increasing the use of a cryptic transcription start site, do you know if the proteins or the genes of these that are encoded by these genes are affected, whether the protein levels are different, or its localization, its translation rate or anything is different?
Yes, we do know that, and that's a very interesting. So let me just point out a very important thing here. That is, most of the protein isoforms will not change, and this is mostly because these evolutionary new exons are located in the 5' UTR. However, if we're not talking about evolutionary new exons and we start talking about genes with an EMAT structure, because they have skipped exons and that not evolved recently, those skipped exons can be located anywhere in the gene, so those protein isoforms would be affected. Is that clear? So we have to think of two different parts, the evolutionary new exons and those genes with EMAT structure that didn't evolve recently.
Right. So you actually showed that the transcript levels increase upon higher inclusion of these novel exons and increased usage of the cryptic TSS. Do you actually know about protein levels for these genes? Did you check any of them?
I'm going to show you a little about 2 slides, I guess. A change in protein levels for genes with an EMAT structure.
Okay, great.
Yes. So let me just go directly to that. So in this slide, I just wanted to point out that those genes in an EMAT structure are enriched in neuronal functions. So this really shows that, or kind of like suggests something and a hypothesis that I like, that is that neuronal genes are more flexible genes, and they are usually longer, and so they have more skipped exons, and they have more alternative first exons, and we're just saying here that genes with EMAT structures tend to be also neuronal. And so let's try to answer that questions about if EMATs impacts also protein in protein levels as well. So we see that EMATs affect transcription levels, so affect expression levels in the mRNAs, and that will be translated to an increase in protein levels per se. However, we see that those protein levels are often increased with a higher effect than what would be only expected for an increase in mRNA levels. So we analyze, yeah, you have a question before this?
You can go ahead, and I'll just ask the question once you're done with the slide.
Okay, so we analyze the polysome occupancy of those TSSs located in an EMAT structure. So now we're talking about genes that has an EMAT structure, not those that has exons that recently evolved. And we see that those TSSs in an EMAT structure, so those TSSs are weak, and they are located right upstream and proximal to the position of highly included skip exons. So those TSSs, we see that has higher ribosome occupancy and higher translation efficiency. So this means that the effect of EMATs on transcription can be magnified by another effect on translation efficiency.
Okay, so the question goes for the previous slide for the Flora and Donna data on translation efficiency. Well, no, we're right here. No, sorry, you were on the right slide. So so what you're showing, is this only true for skipped exons or also true for constitutively spliced exons?
Here, I'm only showing what happened with skipped exons. I'm not showing what happened with constitutive exons. Yeah, this only, it's true for skipped exons at the moment.
But you haven't checked what happens.
I haven't checked. No, that's a good question. I don't know the answer. I don't know what happened with constitutive exons, and it's not that easy to do. But I think it's possible. I think I can do that and get back to you all. Okay. So what I've shown you until now is that during evolution, or not only during evolution, but also in those genes that didn't recently evolve, we see that the splicing of internal exons can impact the usage of alternative promoters and regulate the gene expression levels of the host genes. And in particular, I show you that during evolution, the inclusion of evolutionary new exons can be sometimes sufficient to activate cryptic promoters located nearby. And we saw the strongest effects when those cryptic TSSs are located upstream and close to the position of these evolutionary exons within a window of 1 to 5 kbs. And in general, when we're talking about genes with an EMATS structure, we see the strongest effect when there is a highly included skip exon located downstream of a very weak promoter, and when both are located in very high proximity between 1 and 5 kbs. So we were lucky to be highlighted, our work was highlighted by a few previews that I'm showing here. And I just wanted to quote one of them saying that modulating splicing of proximal alternative exons might help to control the output of EMATS-regulated genes whose altered levels of expression contribute to diseases. And this might be achieved using antisense oligos complementary to splice sites or splicing regulatory sequences. So this is one of the key points of our work, where we start thinking of what can EMATS be used for. So talking about the application of our work, we see two main points. So we can use this to downregulate gene expression or upregulate gene expression of genes wherever we see an EMATS structure. So for example, let's say that we want to downregulate gene expression of one particular gene. So one way to achieve this would be to use an antisense morpholino that blocks the inclusion of a skipped exon here that will in turn affect and downregulate the usage of a proximal transcription start site, and this should downregulate gene expression levels. On the other hand, if we want to upregulate gene expression of another gene, we can achieve that by using also small molecules or antisense oligos that will block, for example, a splicing silencer, so that will increase the inclusion of the skipped exon that will in turn activate transcription from an upstream weak TSS that will in turn upregulate gene expression levels. So we can use this for therapeutical purposes. So with that, I think we're getting to the end of the talk. I would like to thank Chris and all the people in the Birch Lab. And this is a while ago. This is a picture of us a while ago. So here it's more a recent picture of us. And maybe, well, I want to thank you for your attention, and I'm happy to answer questions that are also arise during the talk.
Yeah, thank you very much, Ana. That was a very, very nice presentation. Very interesting journal club here. We do have quite a few questions for you. The first one from Samir Adikari, who asks, is there any evidence of altered EMATS in normal physiology or disease?
Yeah, that's a great question. Yes, as I said, we saw that genes that has an EMATS structure are enriched in neuronal functions. So we try to address the possibility of EMATS regulating the expression levels of neuronal genes in particular. And we did found a few examples where EMATS can be activated or downregulating the specific, some specific neuronal isoforms. So for example, we work with the case of G9A, that is a methyltransferase. that has an alternative skip exon that is that increase its inclusion during neuronal differentiation. So we saw that if we decrease or we inhibit this increase in the inclusion of that skipped exon in G9A, we see a downregulation in the usage of the alternative promoters located upstream. And we see a particular stronger effect for that alternative promoter located more nearby. And that particular isoform of G9A, depending on the usage of alternative promoters and the inclusion of the skip exon, is required for neuronal differentiation. So this is just one example on how EMATS can be regulated in neuronal differentiation. However, we only have one example of that, and we need more work on different candidate neuronal genes to address what is the scope of EMATS on neuronal differentiation or neurological diseases.
Right, right. I have another question from Xiaolao Dong, who asks whether you've looked at the correlation between these novel EMATS exons and back splicing or circular RNA formation.
Oh, that's cool. No, I haven't looked into that at all. So yeah, if someone is interested, I would like to talk. Yeah.
Okay, I've got a technical question here. I think it's referring to the luciferase assay. I don't know if you could maybe go back to that slide. So the question is, what is basically the construct here? How long are the introns, and how does that compare to the endogenous locus?
Oh, okay. So yeah, so here what I'm using is the exact sequence of the rat genome from this TSS to, let's say, here. And then I'm using luciferase as the last exon. Okay, so if you remember the original gene has a last exon located here. So I changed the last exon to be a luciferase expressed exon. So in terms of the length, what is more important here is that there are 500 nucleotides of this intron between this TSS and this 3' splicing. And that is important because the proximity between these two elements, it's a key feature of our effects. But yeah, so the sequence is exactly as the sequence in the rat genome from this TSS to up to here. And it's very well conserved in the mouse genome as well.
I have a question from Irene Bush regarding the HNRNP-U chip and qPCR assay. The question is this effect also seen for other genes with enriched HNRNP-U binding sites?
That's fantastic. That's a great question. So we only test the whole thing with this particular gene. And then, so we did focus on PTVP1 because it's a neuronal splicing factor as well. And we see with PTVP1 here, not with HNRNP-U, that the effects of PTVP1 affected genes are might be mediated by an EMATS effect, because we do see that all genes that are affected by PTVP1 have a decrease in exon skipping or an increase in exon skipping, and that correlates with the increase or decrease of usage of a proximal TSS. So we did kind of like do that analysis in a more genome-wide scale for PTVP1, but not that much for HNRP-U.
All right, thank you. Laura Augusto asks, regarding the HNRNP-U experiment, as we're talking about, So RNP2 is a huge complex. So how do you reconcile RH and RNPU binding immediately upstream of the novel 5' splice site EMATS splice site and recruitment of the RNP2 complex?
So the question is how I'm reconstruct HRNPU binding downstream of
So
the.
well, basically because HNRPU binding is going to be, with you say most of the cases within 1 kb of the EMATS exon, but in some cases further up. So how do it makes sense if the novel or cryptic transcription site is near the HNRPU binding site, but what if it isn't?
Yeah, no, that's a good point. So in this case, I can say that HRNPU binds very close to the downstream intron compared to the location of the mouse-specific new exon. So this is the location where HRNPU binds, and we do see that the binding of HRNPU regulates the recruitment levels of TF2F. And what we see here, looking into different regions, we see that the regions most affected by the recruitment of TF2F are the mouse-specific new exon itself with EDC here, with CD being the most important ones, and not that much about more upstream regions. So that's what the experiment is telling us. I mean, I think it's very important for the splicing factors to be recruiting transcription factors locally. But how much locally that has to be, in a genome-wide scale, we see that it should be located in 1 to 5 kbs. And in this particular case, there are 500 nucleotides between this TSS and this exon, and there are just a few more here up to the binding of HRPU. So
Right.
that
the proximity with the cryptic TSS is probably what ends up making it efficient.
I think so. Yeah, I think that's the key part here. But this didn't like really shows what are the limits of the proximity. But we did that more in a genome-wide scale, and that's what we're saying 5 kbs.
Right. Okay. Maybe a few more minutes to ask questions about the implications of the evolution of those EMATS exons, which also comes back to the question that I asked earlier in the talk. So, what do you know about the evolution of these exons? Are they getting more and more frequent as you go up in terms of organismic complexity? Are they also observed in other animals, in reptiles, birds, or even invertebrates?
Yes, of course. That's actually we're trying to see the scope of this evolution new exons in other species. So for this work, we consider 5 species. We use chicken as an outgroup, and we have cow, macaque, rat, and mouse. And those are the species that we consider. So for the scope of this work, I can only talk about that species. But a preliminary work shows that there are a few thousands of evolutionary new exons in human as well. And what we're trying to do is a similar approach, trying to consider more simpler organisms as well. But I don't have that many data on that yet. And so the other question was about the implication of having these evolutionary new exons, right?
Right. It just adds an additional layer of gene regulation to an already very complex system, especially in the nervous system.
Yeah, exactly. That's what I think. So what we know is that it's not really changing the protein sequence. So it's not going to change the protein structure or the protein function. It's just another way of regulate protein and mRNA levels.
Right.
And
I wonder if it would be a way to overcome repression that exists for the expression of these genes in non-neural tissues. There are many mechanisms that exist to prevent the expression of genes that should only be expressed in the nervous system. So perhaps this could just be an additional way for neurons to make sure that they express the right levels of these genes.
absolutely, absolutely. That's a very interesting hypothesis, and I'm with you on that. Yes, I think that so neuronal genes are very flexible genes, and one way to regulate gene expression is to have a specific new layer on top of all other gene regulation steps. This is another new layer that can only be used when there is a skipped exon and a cryptic TSS located nearby, or a weak TSS located nearby. So it's another way to regulate gene expression, and that is very interesting because it will just, without having to change the expression of a transcription factor that can have like major impacts on the cell, it can only just change the expression of particular genes by activating splicing levels.
Right.
So yeah.
Yeah, I think it's a fascinating question, and one that you will probably be investigating in your own group, as you will be joining the faculty at Boston University in the fall. Is that correct?
Yeah, that's correct. I'm very
Well,
excited.
congratulations on the position. So it's going to be very interesting to see what comes out of your new research there.
Yes, yes, absolutely. So I will be starting my own lab on January at Boston University at the biology department. And yes, I'm very excited to continue work on this connections between different RNA processing events and what are the implications for normal cells and disease.
Great. Well, thank you very much, Ana. We're past the hour, well past the hour. Well, thank you for being with us and being very generous with your presentation and your answers. And again, everybody, thank you for being with us and asking interesting questions. We will see you again next month. So May 19th is going to be our next journal club. Thank you all again, and see you next month.
Thank you so much, Matthew. And thank you all for your questions and having me here today.